So for those of you who don't know me, I'm Fernando Casares. People who like me call me Fer, so I will know whether you like my lectures, depending on how you address me. Well done. So I'm a developmental geneticist. I'm someone interested in knowing how organs and organisms develop by analyzing the function of genes. I work mostly with fruit flies, Drosophila melanogaster up here, but for a number of years now, we've also introduced different model organisms in the lab, some other flies, and some other non-flies, like this insect here, which is extremely interesting, but I won't be discussing anything related to its biology. I work in the Andalusian Center for Developmental Biology, the CABD, which is located in Seville, in Spain, southwest of Spain, for those of you who don't know. And it's a beautiful town close to the beach, so sometimes we have lab retreats. So not only Dr. Laga has nice retreats by the seashore, we also do. Okay. And within the institute, we work in a department called Decision Making in Cell Collectives, which self-defines itself. But the problem I would like to discuss with you today is related to the morphology of organisms and morphology of organs, the size and shape of organs and organisms, which is a problem that comes with two sides, which sometimes look contradictory. On the one hand is the diversity of morphology that we find in nature that originates through evolution. And at the same time, we find that within a species, morphology is constant, basically constant, to such an extent that historically morphology has been one of the major traits used to divide or classify living organisms into different entities. These two phenomena happen at two different time scales. On the one hand is the evolution of different form. And that happens by the repetition of ontogeny, of development. So here you have an egg, fertilized egg, giving rise to an adult. This adult will give rise to new organisms and so on and so forth by passing on its genetic material. So this is ontogeny, this is developmental biology. But as this process happens, sometimes there are changes in the genetic material of the gametes of these organisms, and some changes in morphology or function will arise. An accumulation of these over long evolutionary times will give rise to different species. I just wanted to remind you one thing is that even though the part of the organism that does stuff are the somatic part, in this case the flies, the changes that will impact the next generation mostly reside in the genome of the germline, the gametes, eggs and sperm, etc. So I was telling you, evolutionary change happens in really long time scales, those of that of millions of years, and we don't have direct access in most cases to these times, just simply through fossils and paleontology, and sometimes also indirectly through comparisons of sequences, comparative genomics. But as even Charles Darwin recognized, embryology, or what happens during development, is basic to understand not only how organs form with a particular morphology, but how this form changes during evolution. So we will be concerned with time scales that are longer than the ones that, for the most part, we've been discussing during these days, which is a time scale of hours or days, mostly. And I just wanted to bring your attention to this book published in 1977 of the past century by Stephen Jay Gould, which is a really beautiful and interesting account of the historical relationships in biology between ontogeny, which is development, and phylogeny, which is the origin of different forms. But we know that the action happens during development, right? Which is the transformation of these fertilized eggs into different forms throughout the process involving increasing cell number and specialization of these cells that arise into different cell types to form, say, an amphioxus, a frog, or a human being. This is a picture from Ernst Haeckel, who was a great embryologist. So to give you an idea also of the scales we are going to be dealing with, I brought this picture here from an old book by H.G. Wells, his son, and J.S. Huxley, in which you can compare different sizes of full-fledged organisms. Here you have your regular dog. Here you would have like a bumblebee or a bee or a fly. So here you have the fly magnified. You will see that this is a fly and there are fish that are smaller than flies. There are frogs that are smaller than flies. And even though there are cells that can be huge, and here you have one of these examples. This is a protist cell. This is a unicellular organism that would fit inside some really tiny wasps or some other insects. And here you have just simply the leg of a flea. For the most part, the size of an organ or an organism depends on the number of cells. So the larger the organ or the larger the organism, usually is because the organism or the organ has more cells. Okay? So basically what we are going to be dealing with in terms of the control of size in general of organs is controlling cell numbers. So then we are faced with this huge diversity in nature. So the problem is where to start. Right? So perhaps the best way of starting is trying to recognizing major principles or operations during development that will help us understand this complex phenomena. Went from one single cell into many cells with many different fates and functions distributed in space, working together. And perhaps one of the people who first recognized some of these principles was philosopher and writer and also a biologist, Johann Wolfgang von Goethe, a German writer, who had this intuition that plants, as we know them, are basically variation of a common theme. He thought that basically all the organs of a plant were variations of leaves, okay, the major photosynthetic organs in plants. And that's why, for example, the evolution by artificial breeders of roses, for example, have made use of this fact that the plant is made by modified leaves. So they've been able to breed these roses from wild roses by transforming these stamens in there, these little rods that produce pollen grains. They've been transformed into petals. So that's why this thing came about. So Goethe recognized, therefore, several important principles in building an organism such as a plant. First of all, he would imagine that there's growth, an axis of growth. There would be also a process generating leaves. There would be some sort of periodicity in this organization. So there would be sometime involved some periodic generation of leaves. And then there would be some sort of counting mechanism that will allow, therefore, to order in space these different leaves so that they will be assigned different fates. So, for example, in a flower, you would have the sepals, the petals, then the stamens, and finally the carpels of the female. So that would be equivalent to 1, 2, 3, and 4 in my drawing. So indeed, work during the past 30 years or so have actually defined these processes happening in plants. So for example, here you have a top view of the tip of a growing plant where a flower is going to be formed, and all these buds are going to be flowers happening like these branches out. And within this structure, different areas forming the sepals, the petals, etc., will develop because they express different combinations of genes, generating this sort of numbering system. This also happens in animals. This modular way of developing is very easily seen in most animals, in ourselves as well. But here you have these arthropods. This is a centipede, where you have that the body is formed by a repetition of segments. In this case, the segments, all of them look very much alike. But for example, in this other arthropod, which is an insect, each segment will be extremely different, like short legs or very long jumping legs. Some of the thoracic segments will develop wings, some others won't. The abdomen won't have neither legs or wings. So this is a really powerful way of developing organisms because each of these segments could potentially be modified independently. Of course, you must remember that even though you have this modularity in development, the whole organism is the one that is subject to selection. It doesn't really, it is of no use modifying this segment to make strong legs here if the whole function of the organism is not fit. So this works again, and we've been seeing this before. You have an embryo, and this embryo becomes segmented. That would be step one. The other is each of these segments will have a set of organs, like, for example, a dorsal organ and a ventral organ that might, for example, form a wing or a leg. In all of them, and then a counting mechanism that will tell each segment where it lies relative to the others along the anterior posterior body axis, for example. Right? So, for example, in this organism, if you are an insect, this is basically what you would do to make an insect. Like in the thorax, you will have a pair of wings, 3 pairs of legs, and in the abdomen, no legs, no wings. So here you have an example of that I was telling you. This is the larva of a beetle. You have the head, the 3 thoracic segments, and you can number them, 1, 2, 3, 4, 5. Each of them will be different. Abdominal segments are less distinct among themselves than compared to the anterior ones, but they are different. So in this particular organism, a mutation has been induced that eliminates the function of 2 genes. So every cell in this organism, this is the mutant organism, will be lacking these 2 genes. And yet, the only effect that you see is in this region here, which is the abdomen. Now, instead of having 1, 2, 3, 4, 5, 6, we will have 1, 2, 3, and a repetition of 3, 3, 3, 3, 3, 3, 3, 3, towards the tip of the animal. The one important thing here is that even though all the cells making this organism are genetically identical, you will only see the phenotype happening in the abdomen. And why is that? We've been discussing that once and again, and it's because these 2 genes, the genes that have been mutated in this particular case, are only expressed in the abdomen. And why is that? Because these genes have specific regulatory elements that are able to initiate transcription in this region, but not in other places. So that's why, even though all the cells are mutant, you only see a phenotype in the abdomen, a phenotype which is a change in morphology.
Sorry. So it's all still 333, but how does it know to regulate the shape? I mean, the sizes are still the same as the wild type. So even though you messed around with the gene expression pattern, why is it that the form hasn't changed?
Why these one? You mean these? Okay.
For example, look at the size as it goes down.
Right. I mean, the genes that are operating in the abdomen are not only these 2, ultrabithorax and abdominal A, but there's another gene that is expressed in these posterior segments, actually in a countergradient manner. So it's very expressed here, weaker, 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 and almost nothing here. So even though these whole segments have been transformed into something that looks like 3, that's why I wrote 3 star, is because there's this extra other gene that is doing additional things, and that's why the posterior abdomen cells segments, even though they bear legs, they are smaller. Okay. Yes. How is T different from 2 different from 3? These differences are subtle, right? So it might the difference in here might account to, for example, differences in bristles or things like that, or specific sizes here. So we know that these 2 segments become different later on because these segments will develop strong wings, like these sclerotized wings that beetles have, which are called elytra, while this other one will be developing proper wings. Right? So these are subtle differences, but they are different. Okay.
Also an argument. So these functions are like that they are not affected other circuit. Also makes them very useful somewhere else.
Exactly. This is exactly what I don't know if you heard your. I don't remember your name. Sinya is your.
Chandra.
Okay, Chandra. But exactly this is the idea is that there's modular development of parts that can be regulated independently, and these parts develop using certain modules that you can export from one place to another. Again, playing with these 1, 2, 3, 4, 5, 6, 7, 8 code, for example, which in general biologists will know as Hawkes code. Oh yes, excuse me.
I have it. So, the 456 is not developing because of the suppression of ultrabithyroxine abdominase. So there's a suppression for the segments from 456, right?
Well, it's not really a suppression. The segments are there. So it's the morphology of the segment that has changed.
So that is like the activation of the third segment.
Okay. So the idea would be that this is constructed. Perhaps this didn't happen during evolution, but the truth or the idea is that it's like removing layers of genetic control, right? So this organism has a basic body plan or basic genetic machinery that would tend to make all these segments like these ones, basically, like segments with legs and producing wings. But on top of that, there are these other genes that are able to suppress this function in some places. So modifying this basic body plan to generate these other morphologies. It's not that the segment itself is suppressed, but the function of some genes activating those programs that he mentioned before would have otherwise appeared here. Is that answering your question? Okay. Yes? Yeah. Okay. At this point of development, there's no morphogen gradient acting at this range. Okay. But the morphogen gradient, in case in this particular case happened, which I doubt, acted when the embryo was like the distance in between the two legs or something like that. At this length scale, there's no global gradient acting. There will be local gradients acting, right? So everything that you will find in one segment, you will find it repeated in the next segment. That's what happens. It's a modular structure, repeated once and again. It's something like that. It's similar to what Munia briefly described before. I don't know exactly for these centipedes, but most likely what happens is that in early embryos, you will have an early separation between head and some other segments here. And then you will have here a growth zone, right, that during development will grow. You will have the head, one segment. You generate one segment more. This keeps growing, and so on and so forth. Right? So it would be something like an addition of segments, most likely. If that works like trivolume, for example, the beetle.
Hello, sir. My question is that each segment contains the whole genome, right?
Yes.
So it is like we are expressing some genes, then that function or legs are appearing. Means if we'll control some gene which is responsible legs, so at later stage, means later segments, we can also control or means we can modulate like a segment 2. Is it right or?
That's right. That's right. So you might have some code again, like this would be 1, this would be 2, this would be 3. So when you generate this segment here, there would be, together with the segment, the pattern or the program to generate the segment, there will be here one additional gene expressed, right? That would make 3 different from the others, in case that's the case, right? So that's how you generate. There's some sort of counting mechanism that will relate the time at which a segment has been generated with some sort of identity code. In this particular case, it seems that if that numbering exists, it won't affect the morphology, while in these larvae, it does affect morphology. Right, so genes such as ultrabithorax and abdominal A, UBX and ABDA are transcription factors. So just simply a quick reminder for all of you who are not that familiar with transcription factors. These are proteins that will have basically 2 domains. One domain will bind DNA, right? That's the DNA molecule. This will be the DNA binding domain. This DNA binding domain will recognize specific sequences here. Here you have the ATCG right there. So in specific combinations will be recognized. So therefore, they will locate certain positions in the genome, and they will bind there. And although it's not represented here, most transcription factors also have extra domains somewhere, which will interact with other proteins that will regulate chromatin dynamics and also interactions with the promoter. So these interaction between transcription factors and the machinery they bring on top add enhancers, are the triggers that will activate transcription from the promoters Munia was discussing just an hour ago, right? So only whenever you have, wherever and whenever you have these transcription factors, the enhancer will be active. You will have transcription. So we've seen this picture already a number of times. So it's a wild-type a wild-type fly that has on the second thoracic segment is a large piece of tissue. This is the notum, and large wings. While the third thoracic segment, even though it bears legs, it's much smaller. And instead of having wings, they grow these little club-shaped balancing organs that the fly uses to balance, stabilize flight when it flies. So a single mutation in a gene, this UBX, ultrabithorax, results in the transformation of this third thoracic segment into a duplication of the second. Right? So then you have the notum, this big chunk of cuticle, and also the wings. So there's one really interesting thing here about the action of UBX. It's not only it is transforming the identity and the function of the segment, but it's contributing to make a large structure small structure. So UBX in some way is controlling size of organs. So let me go through recent work that has been published about how UBX controls the identity of organs by controlling the size of organs. Right? That's the thing. UBX controls the development of a small organ which, in its absence, would be very large. For that, I have to introduce a little bit of fly biology. So the adult fly is made piecemeal, made by epithelial sacs that float in the larva. So there will be a pair of sacs per structure of the body. So for example, this second thoracic segment derives from one sac of cells, which is a monolayered epithelium that is called, in general, imaginal disc, because it has the overall disc shape, and it's called imaginal because it gives rise to the structures of the adult. Imago in Greek is adult. So here you would have the wing primordium or the wing imaginal disc, and this little thing will give rise to this whole structure, the notum and the wing. And this is an image from the confocal microscope of this epithelium. At this stage, it's slightly folded, but basically this is a flat monolayered epithelium. And you can see that if we mark different cells with different genes, you will see that this layer, this epithelium, is already subdivided into different regions, right? So there's the red region, there's the blue region, there's here, there's a divide that separates the anterior region from the posterior region. It's like a Cartesian grid. The tissue is being subdivided more and more to give rise to different structures. Well, so at the interface between the anterior and posterior cells, which are genetically different, there's the transcription of a BMP2 molecule called DPP, the decapentaplegic, is initiated. So here at this border, this is the transcription of DPP, which means that these cells are producing this molecule. This molecule is a molecule that can diffuse away from the producing cells. And now if you look at not the transcription, not at the production, but at the protein itself, you will see that this type of cells are the ones producing DPP, but these cells are seeing DPP because it is diffusing away from the source. Right? So what you have here is this pattern. So when you plot the distribution of the GFP protein, the TPP protein, this is the distribution that you get. This is the distribution of the decapentaplegic. This is work that has been thoroughly done by the lab of Marcos González Gaitán in collaboration actually with Frank Juliha. So, okay, the distance these molecules will travel can be in a way characterized by this characteristic length lambda that depends on the diffusion coefficient and also the degradation rate of the molecule. That's typical morphogen gradient formation, which, by the way, Vijay and team, you should, it's either K or mu, but settle it down. So K or mu. So I always get confused when I read all these terms in the equations because you physicists keep changing the names. Not this always here, but K is mu, it can be beta, people like gamma as well. So it's the whole Greek alphabet. So, so what do we know about DPP? DPP we know is involved in growth control, right? This is a normal wing, and this is a wing that is mutant for DPP. You can see that the wing almost doesn't grow. Here you have again, just simply to compare the morphology of the small Holtier and the large wing. They are not to scale. It's just simply for you to see. And here you have in the Holtier primordium in red the expression of ultrabithorax, of UBX. All the cells are red. While in the wing, there's almost no cell red. These bands in there are cells that are not going to contribute to the adult. But by and large, the region that is going to give rise to the wing, which is here, doesn't express UBX, right? So work in the Mann lab and in Sanchez Herrero's lab has shown that UBX, a transcription factor, controls directly in all the parameters that are relevant for the diffusion of DPP. They control the production rate of DPP. They also control the diffusion rate of DPP, and they also control its stability, right? And they've gone beyond that. They've actually tested whether UBX is controlling these genes, these elements directly as a transcription factor. And what you have here is just one of these cheap experiments I mentioned one of the first lectures, in which you are able to detect genome-wide where a transcription factor binds, right? And in this particular case is UBX binding, and here they've compared the places where UBX binds in the leg, which is just simply a different binding pattern, and you can see here that in the leg, UBX binds to these places. That's why you get these peaks here. But in the whole here, UBX, in addition to binding there, it binds to new regions, which are new enhancers, that will control the expression, in this particular case, of thickenins, the receptor for DPP. And they show that UBX controls not only the receptor, it controls elements in the DPP pathway, etc. So here's where I would like to talk a little bit about signaling pathways, right? Because here we are constantly talking about signals being sent and cells reading the amount of signal to do different stuff. So I'm going to describe, yes? The what? Excuse me? The leg. Right, right. It does. But in that particular case, UBX controls very minor aspects of leg development. Right? So the leg in the fly, the leg, a third leg devoid of UBX will look like very much like the second thoracic leg, except for some other things. So UBX controls many things, but it does control different things in different organs. Right? Okay. So you have DPP, which is the ligand, which is binding to a receptor, this molecule here. This would be typically on the membrane. And that binding will lead to a biochemical cascade activating certain molecules. And in the end, this would lead to the regulation of target genes. Right? So far, so good. So you could imagine that now regulates targets like that. However, in the case of the DPP or BMP2 pathway, and I think that Tim alluded to this yesterday, there are interesting targets coming up here. So among the targets, there's a molecule called DAD, which actually inhibits the signaling pathway. This is a negative feedback. The consequence of this feedback is that over time, in a given cell that receives a certain amount of ligand, the signal, which you might imagine that this is the signal here, it transduces. The signal that the cell receives will go like that, but then stabilize, right? By virtue of this negative feedback. So the ligand binds, activates, but then activates that will modulate the pathway negatively. But that would be the one single cell that is exposed to this DPP gradient is seen over time, right? That would be it. However, the things get much more interesting because among the target genes, the receptor is. So now, this signaling pathway will repress the receptor. That means that initially, this cell that is receiving DPP will have this amount of receptor, but after a while, it will have less receptor. So this is very interesting because of the following. So when you have a cell has many receptors, captures a lot of ligand, leaving very little ligand for the next cell. But if after a certain time, this cell represses the receptor, the ligand will continue further down the road. So therefore, these signaling pathways are extremely dynamic, both in time and space. And by doing these sort of things, the shape of the gradient actually will be changing over time as cells respond. Right? So these gradients usually are not static, but are dynamic. And they might be dynamic precisely because of that. Receptors trap the ligand, and if the amount of receptor that a cell expresses goes up or goes down, that will also affect the shape of the gradient. Is that clear? I guess that Tim will explain that again more thoroughly and quantitatively, and you will get it. All right. So things get very interesting when you start like considering signaling pathways as dynamical systems, both in time and space. And these things are really, really important when cells have to evaluate where they are within a tissue and what they have to do as development proceeds. The fact that developmental processes are based on dynamical systems was something that is not new. So one of the precursors of this sort of looking at biology in this way was this guy here, Ludwig von Bertalanffy, an Austrian biologist and mathematician. So if you don't know his work from the 50s, it's called General Systems Theory. So this was a really ambitious man. He planned for a huge research program, general system theory. And at the same time, geneticists were recognizing the generative power of mutations. And a number of geneticists, including Bateson, Huxley, Haldane, Wallington, Goldschmidt, Maynard Smith, were major figures saying, okay, genes must be doing something within this framework. But a number of years had to pass until some researchers, including Perry Alberg in Berkeley and Brian Goodwin in the UK, started looking at the development of organisms as the result of a complex system. Okay. Complex systems in which there are many parts interacting, and these parts are not only genes, but as we have been stressing during the first days of the preschool, genes modify the properties of cells, and this will change tissue geometry, and all these things will generate forces, and all of them will be feeding back on one another dynamically. So the development of an organism as a complex system is perhaps an idea that was best formulated by these guys and some others during the 80s. And if you haven't read this book, How the Leopard Changed Its Spots, it's a great, really great book by Brian Goodwin. And it's a great introduction not only to this idea, but also to the basic concepts behind. So these guys put forward slightly different perspective on how development is controlled. Before them, most people would say that genes are the dominating agents. That was a neo-Darwinian revival of the evolutionary theory by Darwin. But this group of people argued that there were some structural features in these systems that determined the output. Right? As complex systems, the relationship between all these parts would have a structure and will constrain the morphologies that would arise later on. Not everything can happen. Right? And one, in principle, could predict what's the outcome of development by knowing these systems as complex systems. So this slide represents basically this type of thought. This is extracted from one of Brian Goodwin's papers. And in this scheme, very much like Baum tried to do yesterday, he's trying to map the genotype into the phenotype. Right? And here I gave you 3 examples of flies with different head shapes as examples of different phenotypes. And the interesting thing, I believe, is that all these gene frequencies, or basically alleles which are in the genome, are used by the organism as parameters that control these processes. Right? So a subset of these parameters in this parameter space will give rise to a cluster of related morphologies. So by changing these alleles to some others, you will follow into a different subset of parameters that will give rise to a different morphology or sets of morphologies. Right? So these would be the phenotypes or the potential phenotypes. This would be the possible morphospace. And then natural selection would act negatively on these phenotypes to only allow the development of the actual morphospace, those morphologies that can actually work under certain circumstances. So remember, and I think that the artificial life experiment that Baum showed us yesterday, it illustrated very well how natural selection works as a negative force. It decides not to keep certain things that have been produced, but the generative force, the creative force, is in something that is not actually present in Brian Goodwin's diagram, which are the developmental processes. Right? These activities of cells, tissues, that will use these developmental parameters, which are alleles of genes, to generate the different phenotypes. But the whole idea, I believe, of developmental biology is try to go from here to here by understanding how these developmental processes operate. And which developmental processes? Again, yesterday, the artificial life experiment by Buzz was a great example in which he equated certain genes with certain operations, like, for example, cell division, cell movement, or cell death. In his experiment was a direct translation, right? So there was no physical cell there to do the translation. This same sort of approach has been taken by Stuart Newman, who has been trying to classify developmental operations as different units. And following what you were saying before, it's basically like modules that organisms or organs can work with to do stuff. Like, for example, you can migrate and then proliferate, or you can proliferate first and then migrate, stuff like that. So you have some modules that you have to organize in order to generate a particular morphology. And people like Isaac Salazar Ciudad have taken this concept a bit further, and he and his co-workers have developed software like this, they call it Embryo Maker, in which they have realistic, in a way, computational models in which they can where they can actually apply these different developmental modules in order to generate non-trivial morphologies. They allow their epithelium, say, to proliferate and deform, some cells migrate, etc., in given orders, and they examine to what extent that generates some sort of non-trivial morphologies. But even so, the definition of a morphospace, which is the set of morphologies, or the set of morphologies that a developmental process can generate is difficult, because it will depend largely also on the environment, and we don't know how many environments the developmental process can face. Right? So this is a nice example in which you have, these are maggots of a butterfly that have been growing in different environments, and actually this one that is dark and this one that is green become dark and light green just simply because they are able to perceive the background light color around them. It's not what they eat, it's the type of light that they see while they are growing that will change their pigmentation pattern. So even defining what's the potential morphospace of a patterning generating algorithm is not trivial. Sure.
The change of
Okay.
surface morphology of the
Right. So I'm not totally sure, but I think that this is not a structural color. It's not one that you get by changing the structure of the surface, but it's a pigmentation change, and as such, it will require some changes in gene expression. So therefore, the whole picture should look more like that. So it would be this is the basically important thing that we want to deal with. And they would use genes and environment to generate a series of discrete sets of phenotypes that then will be selected, passing through the sieve of natural selection. Okay, so, great. So what I want to do now is to show you a little example from our own work. That has to do with basically trying to understand how a certain morphospace is generated. And again, with morphospace, I mean the set of all the morphologies possible, right? To understand not only one morphology, but to understand the process that can generate all the morphologies and actually define which are the limits of that morphospace, which are the morphologies that you cannot get, because the way the system is built. And this is you will see, is a very simple system. But for that, I need to start introducing the fruit fly eyes, which is basically the same eyes that most insects have. So here you have a fly that is expressing a fluorescent protein in its photoreceptors. So that's why the lateral compound eyes are glowing in red. But also, insects have these three little eyes up there, which are called ocelli. Those are also small eyes. Right? This configuration of compound eyes and ocelli is very old. Okay? You can find it in Odaraya, which is basically something like a crustacean, which was hunting in the oceans like 500 million years ago. Like the wing and the thorax, the head also derives from an imaginal disc, which is called in this case, the head imaginal disc, or the head primordium, also the eye antenna imaginal disc, because it will give rise to the antenna of the fly and the eyes of the fly among the head and the ocelli. And this is an image of this head primordium. Again, you're looking at it from the top, and this region over here will give rise to the ocelli, and this big region over here will give rise to the compound eye. So we will see more about the compound eye tomorrow. But okay, just simply, you have to now to focus on this region here. You see this patch of green cells? So this patch is this one at higher magnification. That's producing another signaling molecule called hedgehog. Again, Tim alluded to hedgehog yesterday while he was talking about sonic hedgehog. So hedgehog is produced here, and you can see these little spots around. So this hedgehog dispersing away from the producing region. This region will give rise to the anterior ocellus, and this one to the posterior ocellus. So these are going to be the two retinas, one anterior and the other posterior. Each of the ocelli, just simply for you to have an idea of the anatomy, will have a large lens on top, small retina behind, and this small retina will be sending axons towards the brain. It is a very old drawing by Ramon Cajal, a Spanish neuroscientist. And here you have again the real stuff. So these are the hedgehog producing cells, okay, these guys here. And these cells are receiving hedgehog. And here what this profile has measured, or is measuring, is how the amount of hedgehog that these cells are seeing, or might be seen, decays in space. Okay? This again, this is if hedgehog is a morphogen, this is the pattern of dispersion decay that you are seeing in real life. And hedgehog is acting on both sides just to specify these regions here with this red gene called eye absent, which basically the only thing that this is telling you is that these cells are going to become eyes, are going to become the retina. Okay? Where this region here is going to form just the region between the ocelli. Again, here you have a high magnification view from the top of the posterior ocellus, the anterior ocellus, and the region in between. So this would be red, this would be the green region, and this would be half of this other red region. You get it? So since these structures are made by two parts, what happens is that the left eye primordium and the right eye primordium will be meet and fuse along the midline, and this would be the left posterior ocellus, this would be the right posterior ocellus, and these two halves will fuse to make the anterior ocellus. You see the topology, or the geometry of this thing. And the morphology I will be trying to understand is the size of the ocellus and the distance separating the two ocelli. That would be defining my morphology. How large the ocelli are and how separated they are. So we know, and this is just simply genetic manipulation, that hedgehog plays a major role here. So these are wild-type ocelli. So these are ocelli in which we blocked the reception of hedgehog, so basically no ocelli. And when we increase the amount of hedgehog these cells are receiving, you will see that the retina are larger. Okay, so hedgehog is controlling not only the development of the ocelli, but is controlling the size of the ocelli. So here we go with another signal transaction pathway. I'm going to get all my colors. Again, here you have a ligand, hedgehog. Then you have a receptor, and this particular case is patched. Then you have a signal transaction pathway, and then some targets. Well, in this particular case, one of the targets is actually patched itself. And in this case, the regulation is positive. Right? In this case, the regulation is positive. But what does this mean? Is that if you have a source of hedgehog, here you have cells receiving hedgehog, and all of them initially have the same amount of hedgehog, the same amount of the receptor, like, say, 2 molecules of receptor per cell. These guys up here will receive lots of hedgehog, and will produce lots of the receptor, which actually will capture a lot. That means that the gradient will change in shape. Why? Because these guys are keeping most of the hedgehog. Right? So again, the dynamics of this signaling pathway in time, for each of them, will change also the dynamics of the gradient being formed in space, likely by slightly a different mechanism. Okay, to explain the following, I need to explain a little bit more the effect of these to explain a little bit more about the hedgehog signaling pathway. This is a kind of complicated signaling pathway in which the molecule that transduces the signal, which is called CI, cubitus interruptus, tends to be degraded. And there's a molecule inside, which is called smoothen, that blocks this degradation. But the receptor patched, right here, doesn't like that. And then represses smoothen. And therefore, if there's no ligand, CI gets degraded. But the binding of hedgehog to patch represses the action of patch on smoothen. So by repressing this repressor, hedgehog acts as an activator. And then when we have hedgehog, we have CI. And then we have targets. Right? So since we are within a gradient with high hedgehog and low hedgehog here, the amount of CI produced will be different positions along the axis, the x-axis here. So there are targets that will be activated at higher concentrations of signal, and one of them is a molecule called engrailed. Engrailed is a transcription factor and has the weird property of inhibiting the transcription of CI. This is weird. So there are cells that, if they receive high levels of engrailed, will shut off the signaling pathway. And there's another target that is activated by CI, which is ice-absent, which this is basically saying retina. Okay? So the idea that we want to understand here is how hedgehog regulates how many cells express ice-absent and how many cells don't express ice-absent, which are going to be these cells express ice-absent, and these cells in between don't. Right? By the way, Suzanne Eaton was the person who discovered this role of engrailed in repressing CI, which is extremely important in the organization of many organs, including this one. Okay, so this basically, I'm going back. I still have 26 minutes, is that right? This basically what I've written down here, but in a very simplified version. So you have hedgehog, higher levels, lower levels of hedgehog, and then the signaling pathway can activate engrailed or ice-absent, or both perhaps. The thing is that since these two targets have different sensitivity, because engrailed is only activated by the highest levels of hedgehog, the domain of expression of engrailed is much narrower, is in closer domain than ice-absent. So you have here, this is the expression of engrailed within the gradient, this expression of ice-absent. Right? So here is where you have the hedgehog gradient. Engrailed is only expressed in the cells producing hedgehog, and ice-absent is expressed in cells that are receiving hedgehog, even at lower levels. Okay, but as you can see here, the cells that express engrailed don't express ice-absent, and that is easy to understand. And that's easy to understand because once you have engrailed in these cells no longer transduce hedgehog. Right? If they have engrailed, they will shut off the pathway by blocking the production of CI. Right? But then, okay, I will go back here in a moment. So you have now the expression of ice absent, and here we also discovered one interesting property, is that, okay, you have a nice gradient like that, and there's no problem with it. But if you have a real-life gradient, and you've seen the profile, it's something like this, right? So in this region of the gradient, where the average concentrations of the morphogen are very low, it is very difficult to be precise, right? So one way of, this is a general way of increasing precision of gradients, is to having a counter gradient, a gradient that will act negatively on the signaling pathway. You will have something that activates a process, and here something, which is another gradient, which will be repressing this activation. So in the end, you will have some sort of very sharp domain of A activation. Right? And this is something that can be seen not only in our little system, but for example, during the patterning of a neural tube in vertebrates. That is an extremely interesting example in which sonic hedgehog from the ventral side, in order to get to be precise, far away from the production site, is helped by a counter gradient of WINs and BMPs that would repress the outcomes of the hedgehog signaling pathway. Right? So this is below these two signaling pathways. The fight is actually fought, or the battle is fought by two transcription factors. So this counter gradient is the WIN signaling pathway, okay, which is produced all around, and it will activate the expression of a transcription factor called homothorax. And I told you that the hedgehog signaling pathway activates ice absent. Well, these two transcription factors repress each other mutually. Right? So by doing so, the border between these territories, the ice absent expression territory and the non-expressing territory, sharpens. And what you have here is one example, one experiment in which with knockdown, we've reduced the amount of this transcription factor in this whole region, and as you can see, the ocelli become larger. Right? It is because in this particular case, we are eliminating the effects of this counter gradient. So I don't know if you know much about how these manipulations can take place in Drosophila, but for those of you who are not Drosophila people, I think it might be interesting to know how the GAL4 UAS system works, and this is one of the genetic tools that we have in Drosophila that make Drosophila a wonderful organism to work with. So this system, the GAL4 UAS, is a system imported from yeast. Right? So there are no GAL4 protein nor UAS sequences in Drosophila, for example. And in the yeast, there are genes that for their activation, they have this enhancer region, if you want. It's called the upstream activating sequence that is bound by GAL4. And once GAL4 is bound, then transcription starts. That's why it's called UAS activating, the upstream activating sequences. Right? So if you had a fly this fly that were to express in this region here of the head GAL4, nothing would happen. Why? Because GAL4 won't find in the Drosophila genome any sequence like UAS around. It wouldn't bind to any place. So now you imagine that you have a fly, another fly, that has a UAS, it's a transgenic fly. All the cells in this fly will have a construct that contains UAS and, for example, an RNAi against my gene of interest, homothorax. This is the gene I want to knock down. By expressing an RNAi against homothorax, this is the messenger RNA of homothorax, the RNAi will find this by complementarity and trigger its destruction. Okay? So now, this guy is potentially UAS everywhere. UAS homothorax RNA. Now these two flies are crossed. And among the progeny, there will be a progeny that will contain GAL4 expressed in this region, and all the cells will have UAS homothorax RNAi. But because GAL4 is only expressed in this region, now this RNAi will be expressed exclusively here, exclusively there. So that's why this fly, when we look at it, excuse me, we'll only have defects in the ocellar region. It's because we've driven this RNAi right there. And we can do that in all the places of the body we want because we have a huge collection and growing of GAL4 lines that are expressed in different places of the body of the fly. If you want to manipulate gene expression in the fly, you can use this technique, and basically you can do it at will. But of course, there's something interesting here. So I told you that engrailed is expressed in cells that receive high amounts of hedgehog, and then they activate engrailed. But since engrailed then shuts off the signaling pathway, then you would imagine seeing engrailed disappearing, right? Because hedgehog activates engrailed shuts off the signaling pathway. So since there's no signaling pathway, engrailed is shut off. Then you don't have the repressor, then the signaling pathway becomes active again. There's engrailed shuts the pathway off. There's no pathway, there's no engrailed, and so on and so forth. But this is not what happens. Engrailed, once it gets expressed, becomes auto-regulated by the action of another signaling pathway, which is delta notch. But in this case, the action of delta notch doesn't require to form an interface. It's just simply some sort of tonic or levels of delta notch signaling. So engrailed becomes auto-regulated. So here, this system describes one general theme of biological organization, like this one, but anyway, which is that we have a signal which might not be very precise at the end, but becomes more precise at the edge because there's a counter gradient that generates different fates depending on where the cells are along the gradient. So this would be A, this would be B. And they're maintained sometimes through positive feedbacks, which is interesting. And some other fates would be maintained by means of toggle switches. And the activity of these toggle switches would be again modulated in space by the combined action of gradients. So because of this region here will express E, while this region quite sharply will express C. That was work mostly done by María Ángeles Domínguez. So I'm going to go over this one because otherwise I won't get to the morphospace. So this is the way we biologists used to represent these genetic interactions. And, what we did next was to spread this out a little bit, just simply to include regulatory points, including DNA. We had a network in which we could see which steps were, for example, just simply biochemical signaling pathways, or which places were regulated by transcriptional regulation. And Daniel Aguilar Hidalgo, a physicist in the lab, went on to model this huge network for these standards using this sort of general formulation in which you have a region where there's transcriptional control, there's a basal transcription from the promoter, and the gene can be transcribed and then translated into protein, and there might be feedbacks, like, for example, positive feedbacks, etc. And he used basically these descriptions of for the process of positive autoregulation, negative autoregulation, or translation. It was very simple. But the whole system was really, really complicated. It was like 13 equations with 61 parameters. So if with 3 parameters, you are able to feed an elephant with 4. But with 5, you can feed my grandmother. So with 61 parameters, you can feed the universe, I guess. I don't know. Anyway, it was a tour de force. He studied that, but he was later on able to reduce the system into a 3-node, basically, network. It's much more manageable. There's no way you can solve this. What Daniel did was he wrote down the equations, and then he analyzed each of the parameters one by one to select those parameters that were sensitive or insensitive. His classification was that, so one by one. So once he had the sensitive parameters, he generated thousands and thousands of vectors of the parameters, and then he basically obtained numerically the simulations, and then he plotted the result. Okay. Still, he did that also for this little 3-node gene regulatory network, in which basically what you have is hedgehog activates this form that will activate CI. Since this is the signal transducer, this form will be activated, so there's a positive feedback loop here. Or you can activate Engrail, and Engrail basically represses these 2 components. And there will be this auto-regulatory term that depends on delta. So that's the basic structure of. There are a number of simplifications. It means that CI, we will say that the system is able to activate the retina, which is basically this thing here, above a certain threshold of CI signaling. Some simplifications, but basically this equation gives you 2 outputs. One is the length of the region expressed in CI in the tissue, which would correspond basically to the width of the retina, and the length of the engraved expressing region that will give you an idea of the length between the ocelli. So this is extremely simple, again, extremely simple morphology that I'm describing here, but basically captures the morphology of the ocelli in flies. The ocellar size and the interocellar distance. So, okay, let me show you the results of Daniel's experiment, computational experiment. What he got is that when he simulated many of these morphologies, again, just simply, I was CI and engrailed, what he found is that the morphologies he would get are not distributed homogeneously.
What are the axes?
Yes, again, I'm explaining that. This is the, if this were 0 right there, this would be the difference between, say, one control value in terms of increasing the size of the ocellus would be like that, decreasing the size of the ocellus would be like that. This size is CI, and this here would be increasing or decreasing the distance in between the ocelli. So that's the. So I'm going to give you here an example. If this were a control value, this would be that value here would be a larger ocelli with larger interocellular distances. For example, this one here would be larger ocelli with the same interocellular distances is predicted not to appear. So only certain morphologies can be found, can be generated by the algorithm. The interesting thing is that in nature, you find that ocellular morphology, and when I insist, when we talk about ocellular morphology, we are talking about the combination of the size of the ocellus and the distance between the ocelli can vary in different species. Right? Here you have two different species, Drosophila species, at the same magnification looking at the ocelli and the interocellular distances. What I did is, okay, this is just simply the product, the prediction of one very simplistic model. But does it capture nature? So for that, what I did was I went to the Natural History Museum in London and spent like 2 weeks taking pictures of all sorts of flies across the whole phylogeny of Hyodiptera. And this is the result. So this is the map of distributions predicted by the model. Again, ocellular size and interocellular size or length. And the dots you see here are the measurements I obtained from fly specimens, from different fly species. And it was pretty satisfactory to see that the trends that I got actually matched this butterfly wing shape that the model had predicted. Especially, there were not too many specimens that fell outside this shape. I mean, this one is very interesting, but that would mean that these guys in here would have basically no ocelli, and the flies I actually photographed had ocelli. So that's why there are no samples, biological samples here. But this to me was an extremely satisfactory result, because we have figured out gene network basically using genetics. We have modeled that, trying to capture the basic regulatory relationships and modeled it in such a way that we made a prediction about the morphospace. That was the synthetic morphospace or the predicted morphospace. And when we looked at the morphospace in real life, we see that it basically corresponds. So this is not the whole morphospace. This is the morphospace of 90 species, but covering a whole range of families. And we believe that finding this match is not random. So I think that we've been able to capture a snippet of the logic of controlling morphology in a Yeah, the point Munia is making is, to what extent I know that this fly and that fly are using the same gene regulatory network. And the thing is that we are assuming that it is the same. We know that basically the same genes controlling the development of eyes are conserved much further away than in between Drosophila. So we are making that assumption, but that's true. That's only an assumption. We haven't looked into very divergent flies and see whether these guys do have the same gene regulatory network. That would be very difficult to do, but yeah, we haven't checked. No, it's not totally right. It depended a little bit on the quality of the samples I got in the Natural History Museum. But for some species, I would have more than one specimen photographed. I don't have them colored here, but usually when you have, like, if I had 3 specimens of the same species, those specimens would be here, like all of them clustered. So the discrepancies in morphology between species are very small. Excuse me, within a species. Excuse me. Okay. Okay, we haven't finished this analysis, but we know exactly where each of the species falls within the phylogeny. We don't know whether there's any sort of scaling relationship or some. We don't know that. It could be done. Yes.
This node, which the hedgehog and
Yes.
they are inputs, right? And the 3 nodes that you are the dynamics of ones that are the 2 on the 3 edges of the triangle.
Yeah.
Nodes of the triangle. Now, so presumably this modeling is modeling in which doesn't have space in it. Where does space come into it? I mean, are you assuming that hedgehog has a gradient?
Yes.
And therefore, you will model the three-node thing, which is probably, correct me if I'm wrong, that's just ordinary differential equations and no space. Right?
No space.
But then the amount of input of hedgehog depends upon space, and that's how you are talking about size. Right? Is that
correctly?
Right?
That's correct.
And now, so therefore, basically, the gradient or the profile of hedgehog is an assumption. Spatial profile of hedgehog is an assumption that goes into the modeling of that. Now, if that is an assumption, then how do you, if that's being put into the model, then how are you getting out these, sort of? I'm a bit confused about how
Yes.
you get these length scales, right? These
No.
are two length scales that you're plotting here.
Exactly. So this as you're saying, this is a given. So
Yes.
so we are just simply drawing a hedgehog gradient. So it might the shape of the gradient might affect the proportions of these things. It might change the but it won't change the overall patterns that we are seeing. Right. So here we used a realistic gradient because we had data about the gradient shape. So we used an exponential gradient fitted to experimental data. So this realistic. But if imagine that instead of having a gradient that is like that, it would be a gradient like that. That would basically change a little bit these relationships, but the general shape of this would be maintained. You would have a concentration here, 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 and not in between, even though the distribution along this axis might actually be deformed.
Yes. What's confusing me is that, you also had that wing thing, that counter gradient that was
Yes.
coming from the other side. And wouldn't that have been another input to the model, which determines the locations of things and sizes of things?
This, if this gradient were totally deterministic with no noise, it would actually be able to convey all the information that we want here, right? So that other counter gradient comes in place when you have a real hedgehog gradient here with a tail that is noisier. So in order to determine exactly where the border between retina and non-retina tissue, you need that level of precision. You need that gradient. But later on we added noise to it. But basically, this is a deterministic model. We don't need that. We can change the shape of hedgehog as we want, and then put a threshold above which this activation of CI will be effective.
So, these curves which you have on this morphospace,
right?
This is your morphospace, right?
Yes.
So the populated points of the morphospace tell you that if the size of the cella is a certain amount, then the spacing between the two must also be related to that. It's correlated. These are correlated. So how exactly are these correlations arising from this model? Maybe does it have a simple explanation, or I'm still not completely clear. The correlations between two spatial sizes is arising from this model, which is a purely temporal model, albeit an input of the hedgehog, spatial distribution. Is there a simple explanation?
I could tell you that Engrade and CI are related through these regulations. That might actually be one of the major causes of the correlation between the two. But if you want, we can go back to the equations but I don't have a ready-made or explanation for it. Yes. Okay. When we did the study, we classified the parameters in 3 classes depending on their effect on on the pattern. So varying, say, 1%, what was the effect on the, by taking them individually. So we classify them into these three. That's the, so therefore, yes, you're right. There are a group of parameters, I don't remember exactly which of them, which tend to give rise to solutions up here, while some others, while they won't do it in here. But I don't remember now which parameters are we talking about here. They were just artificially classified in 3 bins. So, yeah, in a way, we've come up with our little developmental process to explain the morphospace of ocelli, which was something really important for me to be able to have something that will generate not a particular morphology, but it could generate a range of morphologies. I'm finishing now. Just simply, I'm going to leave you for lunch with some videos, high-speed videos of a wild-type fly that is seen a lumen object that is going from top to bottom, and that lumen object is going to be detected by the fly, and the fly is going to have a quick escape response. And you will see what happens. That fly has intact ocelli. It jumps in any direction and stabilizes flight, and you will see it coming out with a very stable flight. So we decided to block the ocelli by painting the ocelli with black ink. Oh, the object is coming and it's not jumping. Hey, jump now. It jumps. Come on, stabilize. Stabilize. Oh, poor guy. Don't stabilize. So perhaps the ocelli work as flight stabilizing things. I don't know where I left. Okay. So just one little thing just before we go for lunch is that in our unit, and I'm using Wednesday to advertise instead of leaving it for Friday, we have through this PhD program, it's called Infinite. We're having a number of open PhD positions, some of which are for physicists. And in any case, I thought you might be interested in checking out our webpage, seeing what our groups do, and then going to this page and find what our groups are offering in terms of PhD programs for those of you who might be interested. That was it, and thank you very much.